The introduction, Erin. My name is Fabrizio Sergi. I'm an associate professor at the University of Delaware. I work in the biomedical engineering, and I have an appointment in the mechanical engineering department. And today I will introduce you to our pilot project that started just a few months ago, funded by the IMBRI program. And the title of the project is Simultaneous TMS and fMRI to study causality of neuromuscular pathways. So I'll start with some motivation and into why it would be important to be able to identify contribution of secondary motor pathways. So in many neuromotor conditions, like primarily stroke, but traumatic brain injury and other other conditions that originate the upper motor neuron syndrome, there's a problem in the corticospinal tract, right? Corticospinal tract connecting, cortical areas to muscles, to motor neurons that, activate muscles. So if the corticospinal tract, the question is, can other pathways take over and support motor function? There are other secondary motor pathways like the reticulospinal tract, vestibular spinal tract, and also the rubrospinal tract that originate from subcortical or brainstem areas. However, progress in this line of research is limited by lack of methods to image function of these secondary motor pathways. Secondary motor pathways that originate in nuclei in the brainstem, like the reticular formation. So the reticular formation, which is the area that originates the reticulospinal tract, we think that he is involved in processing information from stretch reflexes. When you stretch a muscle group, you elicit responses at different time scales, spinal responses that have a very short latency in the order of 30 to 50 milliseconds. And then there are longer responses that involve supraspinal pathways. The longest and the more sophisticated is the voluntary contraction, voluntary response. But the long latency response has a response time between short latency and the voluntary response, between 50 and 100 milliseconds for upper limb muscles. And these responses are generated by supraspinal circuit after integration of information across multiple muscles. We've had consistent but indirect evidence about the involvement of the reticular formation in regulating these long latency responses. However, we've been unable to use measurement as a clinical or even an in vivo tool to quantify in vivo function of the reticular formation in humans because we've not been able to measure motor responses of the reticular formation in humans. So what we have done is we have developed an neuromuscular imaging method that we've called StretchfMRI, which we have been using to identify the contribution of subcortical areas associated to these long latency responses. So in this framework, we have a robotic device that is MRI compatible that perturbs the muscles during functional MRI. We use functional MRI to measure contribution of brainstem nuclei, and we include also surface electromyography or EMG to quantify the elicited responses in different muscles. So these are the three main elements of this technique. We've been developing MRI-compatible robots for different applications, and we've also developed new methods to quantify EMG during fMRI, avoiding motion artifacts. So with this technique, and I'm not going to go into the details of this technique, but we've been able essentially to map the muscle-specific representation of different muscle groups. FCR in yellow, red color scale is a flexor muscle of the forearm. ECU is an extensor muscle of the forearm. And we've been able to map the activation of different nuclei in the brainstem associated to specific muscle groups. So we've seen that for flexor, activation is primarily in the right medulla, and for the extensor, activation is in the left pons, and this is for the right arm, and that confirms that the laterality of the reticulospinal tract is not organized in different hemispheres or different sides of the brainstem like it is for the corticospinal tract in the cortex, but there is a flexor-extensor organization that is lateralized differently. So it means that this could be a good target for intervention in post-stroke rehab. And this in part confirms our previous hypothesized model that had been advanced based on measurement on monkeys, direct recording using invasive electrodes. So this technique is, we're very excited about it, but does have some limitations, which every fMRI technique has these limitations. So fMRI cannot provide insight on causality of the relationship between neural function and behavior. So increase in signal that we measure in the reticular formation may not just be due to the fact that the reticular formation is generating these responses, but it could be due to increased excitatory input that the reticular formation receives from the motor cortex. In other words, the reticular formation may also just act as a relay, and so not generating these responses, but just repeating responses under excitatory action of the primary motor cortex. And so to gain a higher level of understanding of the role of the reticular formation in these long latency responses and to address the question of causality, we are planning to combine inhibitory TMS with fMRI to identify the causal role of the reticular formation. And the idea is pretty simple, as shown in this diagram. The idea is by applying inhibition of the action of the primary motor cortex, we can reduce the input from the two reticular areas and identify whether we are still able to long responses, whether it's attenuated, or whether we can still measure increase in signal information. And so our pilot project has two aims. The first is to validate this neurostimulation approach and validate that there is an inhibitory effect of subthreshold TMS on the cortical contribution to long latency responses. And then the second aim is to study the effect of inhibitory formation during functional MRI to establish whether there is a decrease in signal. So this project is enabled by recently an MRI-compatible TMS system. The picture outside of one, and this is what it includes: the MRI robot that applies perturbation. We have surface EMG to quantify the effect of responses, and we have a TMS simulator. And in our preliminary work toward one, we've essentially effect of our inhibitory protocol that I just described looking at this timing diagram in the left. The idea is that we always apply these perturbations. The onset of a perturbation is timed at 0 milliseconds in this diagram. It's a constant velocity perturbation that will stretch one muscle group. In this case, we're stretching the flexors. And we apply TMS slightly before this perturbation is applied, such that there will be a motor evoked response that is shown here whose onset is at TL milliseconds before the perturbation starts. With this method, we are able to elicit a reduction in EMG signal, leveraging on the mechanism of silent period, right? So there is a motor evoked response, and then there is a silent period, a period where the motor neuron is refractory. And we apply a long latency response during, we apply the perturbation such that the long latency component of this response coincides with the silent period. And so these are the data that we've been collected in different conditions. The first chart is when we apply a perturbation and the TMS is off. So this is a condition where we are able to elicit these long latency responses that are visible as increased surface EMG signal in this time window. And in rows 2 to 4, essentially it shows what happens when we apply TMS, subthreshold TMS, or in this case slightly around the threshold TMS, to elicit a long latency, to inhibit the contribution of long latency responses. And you can see that in this participant, these are 10 perturbations per condition. As a function of different latency, we are able to obtain an inhibition of the long latency responses at all these three latencies. Of course, the latency effects are variable across individuals, so we did a group analysis of our first data set, and the results are shown in this chart, where this is the average long latency amplitude measured in the TMS off condition, where there is a perturbation. This is when we are not perturbing, so this is baseline activity, which we use as a negative control so that we can see that we are indeed eliciting long latency responses. And then we have these three latency conditions, and in all three you can see that there is a substantial reduction in the amplitude of long latency responses. The only one that is significant with this number of subjects recruited, I think this is personalized with 8 participants, is the T3, so the longest delay response. It is the condition where the TMS pulse is sent about 75 milliseconds before the onset of the perturbation. This effect was also obtained at the individual subject level. This is the individuals that we have collected so far. We have obtained significant inhibition in 7 out of 10 subjects for the T3 condition, that is probably the most promising one. And ideally, we would like to have inhibition in all participants, right? Because this will be done then in fMRI at the individual subject level. So to see if we can do better in terms of obtaining effects that are consistent at the individual subject level, we're going to test a different amplitude in TMS stimulation amplitude to see if we can obtain more consistent inhibition of long latency responses using this subthreshold TMS paradigm. And that's all I have for now. So we've been working primarily on AIM-1, and we're going to run a new group of participants in AIM-1, and in parallel start playing with the intricacies of running an experiment like this in the MRI scanner, which will involve some development time. We're grateful for the support of the IMBRI that will support this development project, which would be very hard to find support for otherwise, but is very promising in terms of providing really fundamental understanding of the neurophysiology of stretch reflexes and function of secondary motor pathways. Thank you, everyone, and I'm happy to take any questions.